All right, the floor is yours, Newman. You can go ahead.
Okay. Okay, so everyone, my name is Numan and I am from Pakistan. I have done my master's also from China, from Chinese Academy of Sciences, and currently I'm doing my PhD, it's my first year. And let me first talk about this is my introduction. If ever in the future somebody needs to contact me, I would be available here. Uh, okay, so this is the title basically for my thesis, and I will be sharing my whole research step by step with you about today's Glion GSA Limnoseries and the contents for today's presentation. And let's start with the little words about my master research. So I have conducted research about arbuscular mycorrhizal fungi. Most of your people will be limnologists, so I think they won't be familiar with this term. So basically, it's endophytic fungus. We introduced it and we incubated in the soil, and it can create a symbiotic relationship within the fertilizers, and it can increase the nutrient uptake and the degradation of the grassland or any sort of plants. Basically, it's able to make that symbiotic relationship within the 80 to 90% of plant species. And this is the study area which I have conducted my research. It's in Mongolia, near the Chinese and Mongolian border, and the grassland was Eurasian steppe. And this is me doing some work that I have done the in situ soil incubation. It lasted for around 40 days. And this is the structure. When we have done the microscopic study, we can see those nodes of the fungus. It means they have created a symbiotic relationship within the plant and its roots. And I have recently published this review article related to my master's studies, and these lower two pictures and the bottom is related to my recent study about my master's. And this is the current research article, again from my master study, and it is currently under review. And I would be just sharing the main content, the main finding for the research that the nitrate turns out to be more significant factor in terms of affecting mineralization. And by that, we can be increase the restoration process for grassland. Next, the PhD studies background for my studies. As we know that lakes are very significant landscape for us, holding about 90% of world's fresh water and playing a major role in biodiversity. They also done deal with the burning of carbon, removing it from atmosphere for the longer periods. And currently, I'm working on Lake Taihu, so I would be sharing some stuff with you about this. The Lake Taihu is basically a shallow eutrophic lake, and from a long time, around from 2.5 decades, it has been the eutrophication and cyanobacterial bloom issue. Some recent stuff about the nitrogen pollution. Why is it significant, and why should have focus in limnological parameters about the nitrogen pollution? So it contributes a significant factors worldwide, globally. A typical picture for global nitrogen cycle and how anthropogenic nitrous oxide is affecting our planet and these landscapes. And this scientist you have seen here from Science 2000, he is David Hamilton. He's a very good friend with my supervisor, and he's recently also visited our institute. And the last picture is going for the anatoxin, which is created by cyanobacterial blooms and conducted to be most harmful anatoxin until now. Some recent studies for nature that why it has been a global issue. As you can see in the picture, that in South Asia, Africa, Europe, North America, Australia, and globally, you see the Europe is mostly affected by the cyanobacterial blooms and its eutrophication. And this is the current study for my site about my Lake Tahoe. You can see in the recent years it was fine, but as we are going between 2015 to 16, the blooms were again accumulating, but now it's little decrease by the as compared to the past. Some recent studies, because my study is related to the how the temperature sensitivity and how the nutrients they can increase the rate of denitrification, so we can permanently remove the nitrate from the existing landscape. So these recent studies have been evaluated that the higher temperatures, specifically 25 or 30 degrees Celsius, and high nitrate concentration, they help cyanobacteria to increase the rate of denitrification. And why is it important? This is a like four-step process, mostly done by the Pseudomonas aeruginosa and Theobacillus denitrificus. In the first step, the nitrate go to nitrite, then nitrite, nitrous oxide, and then nitrous oxide to nitric oxide, and at the end, it done to dinitrogen, which is accumulated in the atmosphere. And all of this process is conducted by these specific genes NIRS, NIRK, and NSC clade 1. So even in the recent studies, they have tried to conduct several experiments how the different nitrate reduction pathways like denitrification, NMOX, and DNRA, dissimilatory nitrate reduction to ammonia, all of them, the denitrification turns out to be the best and significant process for the permanent removal of nitrogen. So this is my the first experimental design. I will be taking three different gradients, three different five concentration of three different treatments. You can see five temperatures, five nitrogen doses, and five carbon doses. And we will be doing the in situ ex situ lab experiment. For the nitrogen, I will be using KNO3 potassium nitrate or sodium nitrate, and for the carbon, I will be using the glucose. And this is the complete picture if you can see how we will be able to. So basically, first of all, I take the big sediment columns from the lake, and then afterwards I try to shift them in the small water columns by this pin ejector, and within time they will be incubated for 48 hours. And but the first and tricky thing is I always, whenever I'm conducting this experiment, I need to check the ambient nitrate concentration. It should be around 1.5 or 1. If it's not 1, then I need to pre-incubate, so to create the anoxic and dark environment where there will be no oxygen, so I can add my concentrations according to my desired variables. So here is the details for the experiment, and at the end I also take the surface 2 centimeter for the futuristic DNA analysis. And the one thing is, I would also like to share, I'm not only working for the denitrification, but I'm also working about the greenhouse gases as well, that on different temperatures and on different nutrient concentrations, how the greenhouse gases emissions will affect our expected site. So coming about to some minor results which I have done. So I have done the experiment two times. The first experiment I have done in November 2025 last year, and the next experiment I have done January 2026, just this January. So in the first experiment, some results were good, but some results were awkward. And as you can see in the first picture, the temperature sensitivity give is the result which is well established in the literature as well, that higher temperatures give the best denitrification rates, but the nitrate concentration, the results were fluctuating, like it was not a moderate trends between the treatment. But in the second experiment, hopefully I got the good results, like the lower temperatures are lower, but the higher temperatures and higher nitrate concentrations giving the higher and good denitrification rates. So this experiment was successful for after two attempts, but now I will do it again at a big scale, because Lake Taihu is like a big, it is the first biggest freshwater lake in China, and have area around 2,250 kilometers square. But I will be, this experiment I will be doing now again, but at multiple sites, maybe 3 to 4 sites. And this is the results for the carbon gradient. And you can see in the November and in the January, the treatment for the 25 degrees Celsius is almost the same. I have created this different picture to just to make the complete comparison. On the left side is the last experiment. On the right side, it's a new experiment. But for one temperature, you can see their results are almost similar as compared to before. But when I have done for the second experiment, if I combine that picture, so you can see the results are little fluctuating. In treatment C1 and C2 mean they are giving a bit higher denitrification rates in the 15 degrees Celsius, which is not justified according to the literature, but it has some reasons. Reasons I have mentioned on the bottom, like it could be because of oxygen solubility. Water holds more dissolved oxygen at lower temperatures, and at 15 degrees Celsius, the carbon may remain available longer for denitrification. And the other thing is there were some extreme and outlier readings in my Excel data sheet as well. So this is for now, which I have conducted some experiment and conducted some results. In the future, I will be doing it on big scale. And this is the picture I have created by myself just to indicate and give a bigger picture of nitrate reduction pathways. On the right side, the left side, you can see that when the denitrification is available in a normal range, so the process and shift will be conduct to the denitrification. But if the carbon is highly abundant, like it is available in highly range, so the microbes which are doing the denitrification, they can shift their trends instead of denitrification to the DNRA, dissimilatory nitrate reduction to ammonia. That means the nitrate will be reduced, but the end product will not be dinitrogen, but it will be ammonium. And in this way, the nitrate, it does not permanently remove from the lake ecosystem, but it instead of it stays in our lake ecosystem. And on the left side, you can see the how these microbes and some other zoobenthos, they can affect the nitrogen removal as well as the carbon assimilation. So most of the time, the denitrification process, it has been done by the heterotrophic bacteria because by during the day, they can accumulate the oxygen and create their own glucose. And that's why they can create the blooms as well. This is also the mean data mining from a recent experiment. By random forest analysis, we can see that nitrate again turns out to be the most significant factor in terms of affecting denitrification. And I have created its scatter plot, linear regression, and the PCA analysis of variance, and then heat map with hierarchy to showing the correlation between those all parameters which I was using. I have data for all of these results, but as the presentation time is short, so I'm not able to share all of the results with you, right? And this is some recent zoobenthos studies I have done during a summer internship here. And these specific sites, all of this study which I'm sharing with you right now is specifically from Lake Taihu. Until now, I'm just working there. Maybe in the future, I will conduct some experiments on other lakes as well. You can see the abundance of some bacteria, Skeltonema, Keratella velca, and also here some pictures of zoobenthos, Daphnia magna, Cyclops, Calamoida, and Nereis japonica. It's aquatic worm. And these are some of my recent certifications from UN, from different. I'm also a member of International Society of Limnology, and also I'm a reviewer of two journals, Plant and Soil and Applied Soil Ecology. And thank you.